Thank you. It's a real pleasure to be here to speak in front of all of you. I've been lucky enough to call Lausanne my home for the last 4.5 years, and it's been a real privilege to actually see and be part of the AI scene here, to really see it grow and flourish and be part of that growth. I want to talk to you today about some of the work that we're doing at Isomorphic Labs in building a system of rational drug design using AI. So I grew up, really, before I was in biotech, I grew up in tech. And tech has been dominated by these great exponential charts. You know, we've all seen Moore's law for transistors, and this is something that's persisted for decades now. And if you've seen Jensen's keynote at GTC just in this past week, you know, you'll see this exponential curve for the power of GPUs over the last 10 years or so. And so while things have been really peachy in tech land, where we've been able to come at problems with massive scale and with great breakthroughs in terms of methods development, things in drug discovery haven't been as great. When we look at the FDA and the number of drugs that are approved on an annual basis by the FDA, there's actually somewhere in the order of 40 to 50 drugs on average, and we have 19,000 prescription drug products currently approved for marketing by the FDA. Nevertheless, while it seems like plenty, the disease burden in the world is really still humongous. And so if we look at this figure from our World in Data in 2019, 2.47 billion disability-adjusted life years. Disability-adjusted life years is just a way of saying 1 year of life lost to disease burden. And so if we look, we have all the usual suspects of cardiovascular disease, cancers, other non-communicable diseases, respiratory infections, and so on. And so there's really a lot to be desired. When you look on the right, you actually see the exponential decline of pharma productivity over roughly the same period of time that we've been observing Moore's law. In fact, this has given rise, somewhat humorously, to Eroom's law. And so what we see there as a trend is the cost to create a new drug has doubled roughly every 9 years since 1950. And so the point at which we're at now is that it takes us 13.5 years on average from research to market to bring a new drug. It costs about $3 billion. And despite spending an average of 6 years from inception to when a drug enters clinical trials, 90% of those drug candidates fail, and so there's a lot left on the table. There's a lot of opportunity there. So how can one rationalize over this space, actually? There's a lot that goes on in the drug discovery program, and so I'll share in this talk how we think about different aspects of this today. So I'll walk you through first what the actual sort of drug design pipeline looks like. It all starts on the top left there with target discovery. Before you can treat anything, you need to ideally understand what that disease is. And in modern medicine, we're trying to understand the molecular mechanisms of that disease. What is a particular molecular mechanism that goes wrong? Is there a particular protein that one can identify that if we were, for example, to inhibit that protein or to interact with it, that will actually lead to the alleviation of that disease. Once you know what that target is, there's a process called target validation. You need to perform quite an extensive series of experiments. This can take more than a year to be really sure that that target actually is causally linked to that disease and that it's worth it to go after as a potential drug discovery target. Once you are sure that you're doing this, you enter the hit identification stage, which is a stage where, given your target, you're trying to search basically this massive universe of all possible chemical molecules to find one that will bind to that target, that will basically perturb it in order to be able to affect the disease. When you have a compound that binds to the target, there's actually a lot of different other properties that you want to optimize for that molecule. You want to be able to understand how that molecule will enter the body, how it will be metabolized, how it will be excreted, whether it will be toxic. And so the lead optimization stage helps you do that. After that, you get to preclinical studies where you're trying to basically convince yourself that these molecules will be safe to put into humans. And so you conduct a number of experiments, starting from cell lines all the way through to animal models. And then finally, finishing in clinical trials. You're trying to assess whether your molecule is going to be efficacious. You're trying to assess whether it's going to be toxic. And so you go through this period. When we think about the opportunities for machine learning in this space, there's actually, one could name dozens. But at Isomorphic Labs, one of our tenets is to think about really general problems where if we were to solve that problem, that will allow us to go after potentially hundreds of diseases, that will allow us to reason over chemical space in general. And so bottleneck one for us is this understanding of disease biology. How can we in silico model disease in order to be able to uncover targets better and with higher confidence? Bottleneck two is compound design. The chemical space of what are called drug-like molecules, which is sort of a rough heuristic for molecules that are small enough to make it into cells, is 10 to the power of 60 large. It's a very large space to be searching. Bottleneck three, asserting compound quality. When you're in these preclinical, clinical stages, how can you actually make sure, how can you optimize this experience to make sure that you don't have this 90% failure rate? And then bottleneck four for me is actually exemplary of what technology is best at. When we look at drug discovery now, each program is quite a narrow but deep interrogation of a particular problem in chemistry and biology. And so what happens is that whether that program is successful or fails, it doesn't necessarily scale to other programs. And so if we're able to build models that can generalize, that can reason over this space, we should be able to bring the same type of scalability that have been exemplary of technology. So how do we start rationalizing over modeling in the space? These are molecular systems, biological systems that consist of actually many layers. And when one builds models, you need to think about several different modalities of data. And so these are quantized here into DNA, RNA, protein, interactions between these molecules, cells, and organisms. And you can go much more fine-grained, but I think this is a helpful model to think about it. And then there's a set of methodologies that actually allow you to interrogate these systems. Computational biology is a whole suite of methodologies, of course, that allow you to model these different systems. We have structure-based methods that help us elucidate structure of these molecules. We have methods like molecular dynamics or quantum chemistry that allows us to use physics-based approaches in order to interrogate these systems at atomic or subatomic level. So our first stop on this tour is to look at structure-based methods of proteins. And this is, of course, no incident. As Marcel had mentioned, starting from AlphaFold 2 has been really in kind of the nucleus of how Isomorphic Labs has been started. And so AlphaFold 2 is a machine learning system developed by John Jumper and group at Google DeepMind. Of course, now this is a seminal paper. And the way the model works in inference is very simple. You input a sequence, really a text of an amino acid sequence of a protein. It goes through this transformer-based model and outputs a very accurate prediction of the 3D coordinates of all of the protein atoms. And I would say this paper, when it came out, has taken the world by storm, and we see time and time again amazing applications of AlphaFold. What does this allow us to actually do? When you think about the universe of proteins that have been experimentally determined by techniques like X-ray crystallography or cryo-EM, it's actually a relatively small universe. And so when we look at Protein Data Bank, which is the world's largest collection of such structures, there's 188,000 such structures there today. With AlphaFold, we have been able to make predictions of over 200 million structures, and so these are the structures that one can reference now in the AlphaFold database as of 2024. And this allows us to perform all kinds of incredible work in terms of understanding how very, very complex molecular machines function. And so here on the right, I'm showing you the complete structure of the human nuclear pore, where AlphaFold 2 was used extensively to be able to help elucidate key parts of the structure. And so we can begin to understand the real beauty and the real complexity of how these molecular machines work. But Predicting protein structure in itself doesn't really solve drug discovery. It's not nearly enough. And so what we need to do is we need to be able to bring in other molecules into that picture in order to be able to actually ascertain how can proteins interact with their neighbors in the cell, RNA, DNA, and then crucially, as we design new drugs, small molecules. And so one of the things that we have focused most on over the past 2 years at Isomorphic Labs was in working very closely with our colleagues at Google DeepMind to develop a new system called AlphaFold Latest. And so AlphaFold Latest builds on that technology that has been developed for AlphaFold 2 and allows us to significantly expand the number of entities that we can actually very accurately model. And so you're seeing in this video a few key examples of being able to predict interactions between proteins and ligands, which is really important for us to be able to design molecules and understand how they interact with the target. Be able to predict structures of more complex complexes, protein-protein interactions, protein-antibody interactions, proteins interacting with nucleic acids like RNA and DNA and adding ligands in there. And so how does this system work in a nutshell? It's actually, again, very simple. All you need to do is be able to put in the ligand smiles, which is basically a text representation of the molecule that you're interested in querying, and the amino acid sequence of the protein. You plug it into this model, and what is output is now a joint 3D structure of the protein and ligand, basically considered together in an operation that we talk about as co-folding. And in fact, what you get is not even just one prediction of what that structure looks like, but actually an entire conformational landscape, because of course, many proteins are not rigid at all. They actually have a lot of moving parts, they're changing conformations. Oftentimes when a ligand comes into contact, they will change conformation to open up a binding pocket. And this is actually something that is really crucial to be able to design drugs against some of the hardest targets. So what is the performance of this system? Here's a couple of vignettes. You can also refer to these in the white paper that we have published recently together with Google DeepMind. And so what you're seeing on the left is the performance of the system on protein ligand binding. AlphaFold Latest performance is in the light blue bar on the right side, and it shows quite a significant improvement compared to what state-of-the-art in silico methods are, which are different ways of molecular docking. One key distinguishing factor to understand is that the docking methods actually rely on experimental structure. When you run docking on AlphaFold predictions, you get the bar, the dark blue bar there, and performance is quite dismal. The other thing that docking methods actually need is the definition of a pocket of where to try to put the ligand in. AlphaFold Latest is completely pocket agnostic. And this is, again, very important because in a real drug discovery scenario, you may not know where the binding pocket is, and finding that pocket is a big part of hit identification. On the right side, you're seeing the system's performance on protein-protein interfaces. And while there's quite a significant improvement on the overall protein-protein interface performance compared to current state-of-the-art, there's also a markedly sort of 2x improvement on antibody-protein structure prediction, and this is very important. So, for example, in the bottom right, I'm showing you a predicted structure of the COVID-19 spike protein bound by neutralizing antibodies, and so this opens up very exciting new avenues of research. But being able to quantify, characterize structure is yet not enough for drug discovery. One of the holy grails of drug discovery, especially the hit ID stages, is this problem of predicting binding affinity. So when I have a protein and a ligand interacting, being able to actually predict how strong that binding will be is a key part of hit identification. This is actually what we're trying to do. And so here we have developed novel systems that allow us to very accurately predict what the binding affinity is and actually rank large groups of compounds. When you think about how this is currently done, in absence of these methods, is you do it experimentally. You have something that's known as high-throughput screening. You take millions and millions of molecules and you basically screen them experimentally, looking for a starting point that will have some binding affinity that you will then be looking to expand. You start a chemical series and you try to improve this binding affinity. This is quite an expensive and lengthy stage. And importantly, if actually your hit compound was not in that library, you're not going to find anything, and this is what ends up happening. When you do this in silico, you can actually screen sort of any library you can imagine. And so what we're seeing now in terms of performance is performance of this machine learning system is starting to approach what is called free energy perturbation methods, which are actually physics-based methods where you can basically write down equations and simulate how these molecules will interact. And of course, these are quite accurate, but they're very, very computationally expensive. So what I'm showing you on the right here is just one example of when we performed a blinded study where we looked at both characterizing structure and predicting binding affinity. And so what you see there is a predicted structure of a GPCR. We did not have this structure ahead of time. We only had the amino acid sequence. And then we screened a library of ligands. And what's shown in the different colors there is the clustering of putative binding pockets and poses for these different ligands. And so one can identify several different clusters. And as part of the study, we were able to identify one particular ligand that binds to the allosteric binding pocket that was indeed the result of a fairly lengthy drug discovery effort that has then subsequently been published. Here's another very quick example of these systems in action. So KRAS is a proto-oncogene that is mutated in up to 20% of all cancers and has long been considered undruggable. There's no really easily identified binding pocket for it. And so this has been one of these holy grail problems in drug design. There are now recently molecules that have been put in the market that are actually starting to use very creative approaches called molecular glues to actually attack this target. And so in the study, we're looking whether we can replicate some of the results of this experimental work. The way the glue works is that it covalently attaches to this mutant KRAS, which has 44% prevalence in cancers, and induces proximity with another protein called cyclophilin A. And basically, that attachment prevents KRAS from interacting with BRAF, which is another signaling protein downstream in the pathway that drives some of these cancer pathway processes. So the work we had done there was, again, quite simple. You put in the SMILES of this compound, you put in the amino acid sequences of KRAS and of cyclophilin A, you run inference on this model 250 times, and so what you see in the bottom there is a distribution of different binding poses, and it's actually quite a tight distribution. And we have developed a scoring mechanism that then also gives us a confidence score. As we look at the distribution of these poses, what are the scores that we're most confident in, and also measure that against protein ligand RMSD, which is basically the error in how we're able to reproduce the structure, and you see results that are quite remarkable. There are actually other methods that help us along the way, and these are molecular dynamics and quantum chemistry. I'll speak very quickly now because it looks like time's running low. But here, there is a whole interplay of both physics-based and machine learning methods that actually help us interrogate these systems. So one can imagine, for example, using density functional theory, sort of in the quantum mechanics realm, to generate a whole bunch of data that you can then use to learn a molecular force field, and then use that learned molecular force field to run a series of molecular dynamics simulations. And so in the middle, you see a snapshot of another GPCR being simulated as it's basically stuck through the cellular membrane with a ligand bind into it. And so this gives you very useful outputs of trajectories of how these molecules interact, sort of from a first principles physics approach, as well as predicted binding affinities. And this is very useful both in terms of generating training data for your downstream systems, like being able to predict protein ligand structure or binding affinity, but also for giving you some orthogonal validation for the predictions that these systems may be making. And so it becomes a really rich and comprehensive tool set that you can use. And then finally, I'm bringing into scope this world of computational biology, and we won't have time to talk about it a lot. I'll give you just one small vignette. But there's a whole slew of systems there that one needs to be able to reason over. The genome, the proteome, the cell, the tissue, the organism. How is the human interacting with the environment? There's a set of incredibly rich and complex problems of how one can link someone's genome with their phenotype, what actually we can observe at these different levels. So I'll share just one example here of the work from our sister team at Google DeepMind. Looks like the font there is slightly funky. That citation talks about AlphaFold, alpha missense, which is a paper that's been published in the Science magazine just last year. And so this work basically builds on AlphaFold 2 as a base model. It uses some of the same techniques. For example, this MastMSA training, basically learning to, in the same way that that BERT works, learning to predict a missing residue in this multiple sequence alignment, but then adds this kind of insight, basically this heuristic that when we observe variants that exist in the population, one might assume that the variants that are not in public databases of commonly occurring variants, they are the ones that are pathogenic variants. Of course, this assumption has a lot of false positive signal, but it turns out actually this is really great and provides a great training signal for this model. So you can pre-train it like AlphaFold 2, and then you can fine-tune it using these databases of common variants and basically get a very accurate prediction of variant pathogenicity. And so this takes us to the beginning of this road of being able to say, based on the genome that I have, what will be the effect? This is a very coarse-grained effect. I have this gene, I have this variant, and it's pathogenic, but you could imagine wanting to be able to predict many other things that are acting both at molecular and cellular levels. And I would say this is an extremely exciting research direction, both with us at Isomorphic Labs, as well as out there. So in my last slide, I want to talk about who actually comes together to make these breakthroughs possible. This is a very, very complex field. I started working in healthcare and technology for healthcare about 10 years ago and was very quickly humbled by the depth and complexity of all of the different domains of scientific research and technology that need to come together. And so when we think about Isomorphic Labs, our teams consist of machine learning research scientists and medicinal chemists and DevOps engineers, computational biologists, computational chemists, bioinformatics engineers, molecular simulation researchers, product engineers. It takes really a village to be able to make progress against these age-old problems, and it's been a really great journey for the last couple of years. So I want to leave you by saying thank you very much for your attention, and a big thank you to the team back home and those of you who are here in the audience as well for this great science and for the opportunity to share it with you.
Thank you, Sergei. So this is fantastic, right? It sounds like science fiction, but it isn't. So how you showed in the beginning, right? There's of course an entire pipeline. In the end, you have to do clinical trials and so on. Are you worried that in some sense you won't be able to leverage everything you're doing ahead because there will always be this bottleneck, and we'll just understand more and more and more here, but it just becomes prohibitively expensive to run these clinical trials? What's your thinking there?
I guess it's, do you see it as a challenge or as an opportunity kind of thing? And I think it's very much a challenge. But the great thing is we have great partners along the road. I didn't really talk about our partnerships in this talk, but we have partnered with Novartis and Eli Lilly, for example, to work on a number of programs. And of course, they're amazing at doing this. And so we know that while we continue developing our technologies, we can rely on our partners to help execute some of the later stages that, for example, for us as a smaller startup are, of course, quite difficult. But when I think about the longer term, I think there's amazing opportunities to actually impact clinical trials. I mean, certainly if you're better at predicting things earlier on, you're already going to be better because you should be able to better predict things like toxicity or efficacy. But also crucially, as we get into this computational biology stage, we should be able to define, develop machine learning-based biomarkers to help us better understand the patients that these molecules are going into, and so be able to better understand which patients should respond well to the clinical trials. And so I see this as a key opportunity area.
Opportunity, yeah. And where, I mean, in the pipeline that you're working on, where do you see the biggest bottleneck? I mean, obviously, right, many in the audience work perhaps in companies where there's a serious bottleneck in terms of access to compute. Maybe that's not so much of a problem for you, I don't know. But what if you look ahead sort of for the next 5 years, right? What could be your biggest blocker, and what are you doing about it?
Yeah, that's a great question, Marcel. I mean, the way we see it is, first of all, there's never enough compute.
I hear.
Even if you're part of Alphabet. And so, you know, I think making sure that you have access to resources is going to be always limiting, especially in a field like machine learning and deep learning, where this is such a highly empirical field that you just need to try a lot of stuff before something works. And so access to sufficient compute, I think, is really important. The other one is access to data. Of course, biology data is, you know, ostensibly quite a bit harder to work with than some of the sort of natural language data that we're used to. You can look at a sentence and know if it makes sense or if you like it, how you feel about it. It's not so when you look at the results of an experiment. And so a lot of what we rack our brains over is both what are the good data sets that already exist out there, but also how can we bring that data in, perhaps generate that data experimentally, and how can we actually design these data sets in a way that are going to be most impactful for machine learning. You know, one of the things that we observed is that in drug discovery, maybe life science in general, of course, people generate their data for a particular problem, for a particular study. And so while you may have a large sample size, it doesn't really necessarily hold that that data is going to be great at bolstering the performance of your model. You know, when you build these general models, you want to have diverse data sets. And if you think about the universe that we're modeling, like let's put ourselves into that protein ligand prediction context. You know, we have millions of potential proteins. We have 10 to the power of 60 potential ligands. So any notion of being able to cover that space are, of course, futile. So data set design in order to be able to build these models is something that we think about. And then the last one for me is talent, essentially. How can we bring the most talented, you know, professionals? As I've said in that slide there, you know, there's so many folks you need to bring to bear on this problem. Yeah. How can we actually attract them to this field? And, you know, I found this conference and actually this area in general to be a great melting pot.
Yeah, okay. So maybe in that sense, a quick last question. You're building offices also here in Lausanne in addition to London, and that's of course great for us. What is it that you think, you know, a system, an ecosystem like this one could do to help you and others advance as fast as possible?
Yeah, I mean, I think it's fantastic what's already started here, and we only need to continue strengthening it. You know, when I came here 4.5 years ago, I found, you know, this incredible university, EPFL, with a great balance, I thought, between a focus both on academia, but also understanding that a lot of the folks graduating from here are practitioners that are going out into the industry. And as you've shown in your own figure, you know, the number of industry labs that are actually producing groundbreaking research has just ballooned. And so I found this sort of great ecosystem. And so in my mind, you know, we need to add in those pieces. We need to make sure there's access to hardware, and so I was, you know, really pleased to see that we're making great steps there. We need to generate amazing data, and there's great experimental labs here. You know, we'll call out labs like Bruno Correa's lab, for example, that are doing really inventive experiments that will help us characterize some of the space. And then we need the talent, and that's the students. And so I think it's a great ecosystem to be able to do that in.
Great. Well, we're happy to have you here, and we're looking forward to work together. So thanks very much.
Thanks, Marcel. It's a pleasure.
Pleasure. Yeah, you can leave that here.